And as Rachel mentioned before we got online, I did spend some time with Rachel and Dr. Birgitta Bergman in Stockholm back in a year that I don't want to divulge because it reveals that I was there when I was 5 years old. That's a funny math way of trying to rewrite your age. Never mind. But it was a fantastic and really enriching time. And I think Rachel would agree it probably changed both of our professional careers and certainly changed her personal trajectory as well, as many of you know. So today I'm going to talk about something that I've been working on and interested in a very long time, starting with my postdoc at Princeton, and that is to understand how marine diatoms take up iron. First, I want to thank the U.S. National Science Foundation, the Oceanography branch, and a branch within the Biology Directorate for funding to do this work, as well as collaborators Mark Hildebrand, the late Mark Hildebrand, I'm sad to say, Jeff McQuaid and Andy Allen, as well as students from my laboratory. These are pictures of diatoms. They're very pretty. They're among the first organisms that the dude who invented the microscope looked at after he was done scraping his teeth and being grossed out about the stuff on his teeth. He was fascinated with diatoms. It's just an amazingly beautiful set of organisms to look at because of their glass frustules. And also their glass frustules are preserved in sedimentary records, which makes them amazing proxies for paleoceanographic and paleolimnological conditions. So my general research interests are what governs the fitness of marine phytoplankton. So you're all marine biologists. You know that the base of the food chain, of the food web, I should say, are marine phytoplankton. And diatoms make up a significant chunk of that base of the food web. So I take a reductionist approach. I'm really interested in understanding the mechanisms for acquiring nutrients like iron, carbon, and nitrogen. And today I'm going to focus my discussion on iron. So we use a variety of techniques, some of which are older than even Dr. Foster and I, older than me. I work on my English skills here, and some of them are very new. So the radio tracers have been around since the 1940s and 1950s. Microscopy, classic algal physiology is still very important. And this, coupled with more recent methodologies like reverse genetics and omics, are really important for bringing that whole ensemble of skills to the table is important to answer some of these questions. I should also say that omics approaches, while they're very sexy and very appealing, they can't really be leveraged to their fullest extent without a command of the algal physiology. There's been plenty of examples of omics studies where the physiology has been done poorly and then scientists have been misled. So we try to take a holistic approach looking at all of these, bringing all these tools to bear. So this is an outline of my talk today. We're going to talk about diatoms briefly. Why are diatoms important? Why is iron important? We're going to talk about 3 different pathways of iron uptake. One, we'll briefly mention the non-specific divalent metal transporters, of which a favorite protein of mine called NRAMP is one. Then we'll talk about a pathway very well described in yeast, called the reductive oxidative pathway. And then another pathway that was thought to be restricted to metazoans, and that is the transferrin pathway that operates in our bloodstream to keep iron away from bacteria and to provide it to red blood cells who have a very high demand for iron. So I'm going to talk about what we know about iron physiology in diatoms. I'm going to talk about the era of kinetics. So before we had omics and reverse genetic tools, we relied on doing very tedious and careful experiments at pico to nanomolar levels of iron using radio tracers to decipher the mechanisms of iron uptake. And I'll try, and once I've put you to sleep at that point, I'll try to wake you up again and really tell you about some of the exciting stuff invoking omics and reverse genetics, and then we'll summarize. Who are diatoms? Well, this is the eukaryotic tree of life, at least as of 2003. There's been a few modifications since then. But what you'll notice, let me enable my pointer here. You'll notice diatoms are way on this side of the eukaryotic tree of life. whereas most of what's in our biochemistry textbooks have been written from what we've learned from opisthecons, which includes animals and fungi. Almost half of the National Institutes of Health here in the United States, not half, but a good chunk of them work on fungi like yeast, because the core evolution between humans and yeast is fairly well conserved. So most of what's in our biochemistry textbooks have been written from what we've learned about fungi, animals, and land plants. And you can see that they're very, very, that they couldn't be more unrelated to diatoms within the eukaryotic tree of life. Of course, diatoms are beautiful. These glass shells are what help them also be very important for biogeochemical cycling, which I'll talk about in a minute. But another weird thing about diatoms, and I noticed you had Sarah Smith as one of your guest speakers here, and this is a slide that I borrowed from one of her papers. She's certainly not the one to first describe secondary endosymbiosis by far. Of course, that honor goes back to Lynn Margulis. They are derived from 2 rounds of endosymbiosis, right? And I don't have time to dissect this entire diagram, but these 2 rounds of endosymbiosis, the first round where a cyanobacterium was consumed by another organism forming a eukaryote, a photosynthetic eukaryote, and then another heterotrophic eukaryote came along and consumed this photosynthetic eukaryote. So those are 2 rounds of endosymbiosis, whereas the organism that was consumed was not digested, but rather the genetic material from that organism was incorporated into the host. And in fact, most of the genes end up in the nucleus. So you end up with this weird situation where the proteins might need to go into the chloroplast, but they're encoded in the nuclear genome. So the messenger RNA is made in the cytosol, the proteins are made in the cytosol, and they have to get back into the chloroplast. Very complicated chloroplast with 4 membranes around it instead of the classical 2 membranes you see in land plants. And so there are people who spend their whole careers to decipher how this happens. But this, the way diatoms do business is in part related to this very much of a patchwork of their evolutionary history, right? So they've got genes from their primary endosymbiont, the secondary endosymbiosis event, as well as a lot of genes from horizontal gene transfer. So they're very complicated organisms. So I also do want to take a step back through Earth's history and talk about why iron is so important to life as we know it, especially among eukaryotes. And so this is a log plot. This is some data that I pulled from Max Sato's paper. I noticed he was on your list of speakers as well. And so this is a log scaled plot of oxygen concentration, carbon dioxide, and iron. And up here at the top, this is in billions of years ago, we have key evolutionary events in Earth's history. So what I want to focus on here is that about 2.7 billion years ago, some photosynthetic organisms decided that it would be pretty cool to split water instead of hydrogen sulfide and to release O2 into the water column. And so you'll notice O2 levels back in early Earth's history was a fraction of a percent of the atmosphere. And you all know now that O2 is about 21% of the atmosphere. And so 2.7 billion years ago with a B, these organisms evolved to split water, make oxygen, and it wasn't for another 400 million years that oxygen levels started to creep up. And what happens, I took my marine chemistry slide out of this talk, but iron is very poorly soluble at high pH and high oxygen. And so iron levels in the ocean went from 50 micromolar down to today where the values are less than 1 nanomolar. So you have like a 6 order of magnitude drop in iron concentrations. But at the time that most of the core biochemistry was evolving, iron was the obvious choice as a carrier of electrons. it is redox flexible between iron 2 and iron 3. And so iron was chosen as the choice for most of the biochemical pathways involving electron transport. And so you didn't see a drop in iron, though, for about 400 million years because or a rise in oxygen. The oxygen didn't rise in the atmosphere until it had reacted with sufficient iron in the ocean to create banded iron formations, which you may have heard about. And banded iron formations can be found in many geological provinces on the planet, including wait, who's this young lass? So this is a photograph taken with this old device called a film camera. That will be on the exam. And with this film camera, I was able to document this beautiful picture and banded iron formation here in Biuram, Australia. So we still see relics of this amazing pollution event even today. Okay, so this is the photosynthetic electron transport chain. And what is shown in orange is all the cofactors that require iron to facilitate that electron transfer. I'll speed it up a little bit. You can read this quote by Kipling up here. And so iron really is master of them all when it comes to the trace metals required for life as we know it. Now, in the open ocean today, with the presence of 21% oxygen, iron is very poorly soluble. And there have been a number of iron fertilization experiments in these regions where plant food nitrate is very high, so-called high nutrient, lower than expected chlorophyll or HNLC areas. And this is an older slide. There's been some more of these experiments since then. But in all of these experiments where people have added iron into the ocean, phytoplankton bloom. And diatoms are the principal winners of those experiments. This is a very simple micrograph, again, taken with one of those film cameras attached to a microscope. And you see before iron enrichment, a lot of small algal forms. After iron enrichment, large silicified diatoms tend to be the principal benefactors of iron addition. But not all diatoms are created the same. In fact, it's a very polyphyletic group, very diverse, lots of evidence of horizontal gene transfer, as I've mentioned, kind of superimposed on their very freaky evolutionary pathway with secondary endosymbiosis. So not all diatoms respond to iron the same. This is a field experiment in the Pacific Ocean with 2 different diatom species here. Whoops, there must be some automatic timer on my show, I'm sorry. Before iron addition into the field, the growth rate of these 2 algae were this. And then after iron addition, they both responded, but you see Catastrophes debilis was the clear winner here. So this is field evidence that not all diatoms are created equal. And then in the laboratory, these are 2 diatoms I'll talk about today, the pennate diatom, Phaodactylin tricornutum. This one happens to be genetically engineered with a GFP protein, which is why it's glowing green, and Thalassiosira pseudonana. These are the first 2 diatoms for which we have complete genomes. And what I'll try to focus your attention on here, this is laboratory data, where we look at the growth of the algae versus the picomolar concentrations of available iron. And we see that Phaodactylum can grow at 1.0 per day with 28 picomolar, whereas T. pseudonana requires 170 picomolar to get that same growth rate. So if you mix them together in the open ocean, clearly you could tell who's going to be the winner. You're not going to have T. pseudonana doing very much. Okay, so I'm going to just jump into 3 different pathways. I'll spend a very little time on this first one, the non-specific divalent metal transporters. They include a variety of proteins that you'll come across in the literature, depending on what organisms you're working on, IRT, NRAMP, zinc iron permeases, etc. This is a fascinating study. It's old, but it's still fascinating to me, where they, this group individually, they tweaked individual amino acids, and they showed that they could change the specificity of divalent metal transport. This goes to show that bioinformatic predictions of what some of these proteins do in terms of specificity are very difficult, because we don't know why changing, amino acid number 136 in this chain suddenly made it not take up iron, but start taking up zinc. Okay, The other thing about divalent metal transporters is, well, they're not very specific, and they're not very useful under super low iron concentrations because they're not specific. They'll end up taking up things like cadmium and zinc when the cell really just wants iron. I do want to introduce you to a very well-studied pathway in yeast called the reductive oxidative pathway, or redox pathway. It involves a very peculiar set of reactions where iron is first reduced from iron 3 to iron 2, and then it is concomitantly oxidized back to iron 3 and passed on to this permease shown in green to take up the iron 3 into the cell. Why does the organism go through all this trouble of spending an electron to reduce the iron and then turn around and oxidize it? Well, because this is highly specific. There's no other trace elements that will sneak in through this pathway. Okay, the only one that is even susceptible to reduction is copper, and then the peroxidase is believed to play a role in sparing the cell from copper toxicity by reoxidizing the copper back to copper 2. Okay, so in this pathway, it's mediated by ferric reductase. I'll show that as blue. Reduction is blue. The oxidation is a rust-colored orange, and the permease protein shown as green. And so what we know from the literature in yeast is that the permease cannot get at iron 3 directly from the dissolved milieu or from the seawater. It has to get it from cooperation with the permease. I also want to introduce you to what's happening in your own bloodstream, and that's mediated by a protein called transferrin, which is shown here in these little green dots. It's a heterodimer, and transferrin binds iron in your bloodstream. Once it binds iron, it lands on these receptors on your cells, and that kicks off a series of reactions that involve endocytosis of these receptors, these clathrin-coated pits. And then once these endosomes are inside the cell, it's protonated, which releases the iron, and then the transferrin protein is released back into the bloodstream. This is super effective at keeping bacteria from getting at the iron in your bloodstream, okay? And there's a reason I'm telling you about human physiology here, which you may or may not know about from the literature, but this is one of the most fascinating things that projects I've ever been involved in, and so I'll tell you about that in a few minutes.
Excuse me.
Yes.
So transferrin is a way for our cells to able to like collect iron without bacteria is coming in and like steals it from us, basically.
Yes. So the transferrin binds iron with such amazing specificity. Besides specificity, the affinity, it binds it so tightly that bacteria cannot wrestle it out of transferrin's control. So there's a constant warfare in our bloodstream. And if you're healthy, you don't have bacteria in your bloodstream. So when you're sick and someone does they do a bacterial plating, your blood should be basically sterile if you are not ill. And if they're able to culture bacteria in your blood, then they know that you have a major problem. They'll probably already know because you'll be running a crazy fever.
But does the body increase its production of transferrin when there's like bacteria in the bloodstream?
I don't know. It's possible. That's a good question. I'm not sure. Yeah, I'm not 100% sure. So the transferrins, like I said, it's a heterodimer, and they get together and they're able to bind iron 3, but they also have an anion that is that co-binds, if you will. So it's iron 3 and the carbonate anion, and these seem to have evolved from a periplasmic binding protein that had been involved in phosphate binding in archaea, in ancient archaea. So in humans, this has evolved in metazoans and other animals. This has evolved from this archaeal protein. And so we're going to get back to that in a few minutes. So now I want to tell you a little bit about By the way, I don't have a clock in front of me. How are we doing?
Plenty of times.
Okay. So I want to tell you about some of the stuff that we did before we had genomes of diatoms. And so I want to kind of explain to you what we know about iron uptake in lab cultures. And so this is a plot of a set of experiments where individuals looked at the rate of iron uptake by a marine diatom under 4 different concentrations of total iron in the media. This is the log scale, so 10 to the -8 is 10 nanomolar. And I should mention that the iron is buffered in that media by the presence of aminocarboxylic acids such as EDTA. EDTA is the one that we most often use. NTA, CDTA, and DTPA, different functional groups, have different binding affinities for iron. And so there's an equilibrium reaction with iron EDTA with free iron, Fe3 plus EDTA. And that free iron is also in equilibrium with other species in the media. Collectively, we call all those inorganic species Fe prime. So in this experiment, let's just focus on what's happening at 10 nanomolar. We see that with 10 nanomolar iron, the uptake rates vary by a factor of 100. This is the uptake rate per cell per hour. And so with the same 10 nanomolar iron, you get a 100-fold difference in uptake rate. And so if you're using DTPA, your uptake rate is low. NTA, your uptake rate is high. And what I've done is I've color-coded these here. So you see that the stronger the ligand, okay, which is shown in black, the lower the uptake rate. The weaker the ligand, the higher the uptake rate. Once you calculate the Fe prime value based on that, on these experiments, and you plot your data, your uptake data as a function of Fe prime instead of the total iron, you see instead of this very large disagreement, you see a very consistent agreement, suggesting that Fe prime is the species that's being taken up in culture with EDTA, other aminocarboxylic acids. Now, when we move to the field and look in the ocean, a couple of things started to emerge in the 1990s, and that is that a lot of the iron in the ocean is complexed by organic ligands even stronger than EDTA. And we were able to, through electrochemical methods, break these down into 2 classes of ligands, the so-called L1 and L2 class. And I don't have time to really get into what makes up that whole gamish of compounds, but they're basically operationally defined on how well they bind iron. And so this is a log plot. So we see the total amount of iron in the water is lower than the amount of the ligand. And when you calculate the iron that's available for uptake, it's super, super low. So 0.1, 0.2 picomoles of Fe prime per liter. And that plots out on this plot here. This is some growth rate data from a mentor of mine, Bill Sunda. These are a bunch of different diatoms and a coccolithophore thrown in here for good measure. And we see that the growth rate versus iron relationship looks generally like this. And that the amounts of iron in the ocean are much lower than this relationship. So then it begs the question, how in the world do diatoms grow in the ocean? And so we thought, well, there must be something wrong with our model. And so there is a couple of things that people have suggested, that maybe some of the iron bound to these ligands, some of which consist of siderophores, which are well characterized in soil ecosystems, might be reduced by ferric reductases, and then that newly released iron may be available. So I'll blast through this slide real quick. We developed some pretty sensitive tools using radioisotopes, taking advantage of the differences in the chemistry of EDTA, which is hydrophilic, and ferrozine, which is an iron 2 binding compound, which is very hydrophobic. So we were able to use very low amounts of radioactivity and filter cellular material onto filters to count for particulate uptake, and also to look at the amount of iron 2 in the media using a resin that has 18 carbons called a C18 resin set pack. and that binds the iron ferrozine complexes. It's radioactive. We can then count it very specifically and get very robust amounts of iron 2 production in media. I'm going to tell you really quick in 4 slides about 2 years of work from 2 postdocs, as well as our advisor. So 4 slides, 2 years of work. And so it's a challenge to communicate all of it, but without putting you to sleep. But this is iron uptake rate in a solution where we used a iron siderophore complex called ferrioxamine B. Uptake rate control is shown here in black. We add a lot of siderophore complex that doesn't have iron. We see the uptake rate goes down. That's not the surprise. The surprise is when we added EDTA, the uptake rate went down. And all this jibber jabber up here shows you the conditional stability constants, that the strength at which iron is bound by ferrioxamine B is 9 orders of magnitude greater, I think if my math is correct, than the strength at which EDTA binds the iron. So there's no reason that EDTA should be able to take the iron from the iron siderophore complex. So this gave us pause into what's going on. We also have this other result where we could titrate down the uptake rate with the iron 2 binding chelator. And this led us to suggest, this is one of the components of this new model called the iron 2 cell surface model, where iron is reduced by these a reductase-like moiety, shown here in blue. And then a second step is required for transport, and that a weak ligand like EDTA can actually outcompete iron uptake from a strong ligand like ferrozine. Again, this reaction doesn't happen. So there has to be this 2 sets of reactions. There's like a 30-page paper that we published on this if you're really interested. I keep it by my nightstand for nights when I'm thinking about politics and I can't sleep. So the other part of this model is that this iron 2 binding ligand ferrozine was also able to inhibit uptake, and ferrozine has no business taking up iron from EDTA or from iron 3, and so we argue that the ferrozine is wrestling the iron away from the cell surface. So to summarize that 4 years of work, reduction of both iron siderophore complexes and what I could call inorganic iron, or Fe prime, is critical. It happens in 2 steps. It's a completely kinetically derived model. We wrote the paper before we even had the genomes for the diatom. And so that's why I refer to this as our pre-genomic era information. Now, the first genome was published for T. pseudonana. We were very happy to find that, in fact, there are reductases in that genome. There are divalent metal transporters as well, which I won't really get into too much, but reductases, peroxidases, and permeases. So it's consistent. Our modeling results are consistent with the appearance of genes from T. pseudonana. And when we look at the quantify the transcripts, we see that this is just growth rate versus iron in the cells. We see that the reductase transcripts are upregulated under low iron. Permease transcripts, of which there are 2 in Thalassioscirus pseudonana, also upregulated. But then we got this result with this rust-colored plot here for the peroxidase, where not so much. We don't really know what's going on. And so we said, well, maybe this is just a case of, proteins not being equal to messenger RNA abundance in eukaryotes. So we published this work, and then I went on to my position at Rutgers and started diving into proteomics. So this is some work that my master's student had bravely embarked on, where we use a compound that has these functional groups. This is a biotinylation approach. And so this is a cell surface with the amino acids of a transmembrane protein showing here. And the red amino acids here are lysines, which have a functional group which reacts with this succinimide ester shown here in green. And then this biotin on this molecule can be purified from a complex mixture through a very well-characterized system called the avidin biotin system. So you basically label the outside of your cells, and you then pass it through an Avidin column where only the biotinylated proteins will stick. You wash the rest away, and then you sequence the proteins using mass spectrometry. And so we found 2 permeases in this diatom. So we know that those are expressed. I told you the motivation for the proteomics was the peroxidase. We still have not seen a peroxidase in our proteome. More direct evidence comes from some genetic engineering work that we've done in collaboration with Mark Hildebrand. And in this case, we have modified the diatom's genomes to express the FTR as the permease, and green fluorescent protein is a common marker. And so we've labeled the permease we engineered this protein, ferric permease GFP chimera. Chimera after Greek mythology of, was it half goat, half snake, and half lion, which doesn't make sense because clearly I'm not good at math. And so anyway, so we made these chimeras of FTR1 and FTR2, these 2 permeases. This is some of my graduate students' work. Chlorophyll fluorescence is shown here in red. GFP fluorescence is shown here in green. You see there's a wild type GFP bleed through, which we've been able to mathematically deconvolute. But basically, we see expression of both permeases at the cell surface. And so this really raises a question when we think about the yeast model. This is that canonical yeast model. So canonical, kind of subject to canon law. This is what everybody, believes. So this is the prevailing model that's been, or conventional model, you could say. And we've come up with these 3 other scenarios. How do you have 2 permeases shown here in green? How does that jive, or is that consistent with the model in yeast? And so we've come up with these 3 possible scenarios. I think my favorite scenario is probably the last one, but we're working on testing these scenarios. We're trying to develop ways to, so we basically don't think this peroxidase is involved at all in iron uptake. And so we have the ability to knock down genes, but we don't have any, the ability to knock out is restricted to like 2 or 3 labs in the world. And we're trying to get that going with those labs to further evaluate this pathway. What we do know is from knocking down the expression of the ferric permeases using reverse genetics, we see that the uptake, this is a plot of growth rate suppression. So wild type is suppressed at low iron by about 20%, but both of the knockdowns are suppressed by 50%. So we know these ferric permeases are doing something. Our working hypothesis is that they're actually not behaving like they behave in yeast. If you think back to that eukaryotic tree of life diagram, maybe that's not a surprise once you view it in that context, that diatoms are so different than yeast in terms of their evolutionary history, right? So that's a working project. We think that that these permeases are taking up iron independently of the yeast system well characterized in yeast. Now I want to talk about omics. And I was happy to, I didn't photo, this is not my photograph, but this is somebody throwing spaghetti against the wall. And that's an expression when somebody is doing an experiment, they don't really know what's going to happen, and maybe the hypotheses are poorly defined. But when these omic approaches first started coming out, I was a little bit skeptical. I was like, come on, man, have a hypothesis. Don't just throw all your new fancy, sexy technology at the problem and hope an answer is going to emerge. And a colleague of mine, so make no mistake, I have high respect for, went the omic route, throwing the spaghetti against the wall. And I'm really glad that he did. One of the things, when you have a hypothesis-driven question, you are might only find what you're looking for. Whereas omics, the systems biology approach, I think it's particularly important. I've really come around quite a bit, especially when you talk about the fact that diatoms never got a copy of the most recent biochemistry textbook. Everybody understand what I'm saying? And if they did get a copy, they didn't read it because they said, you know what, I've been doing my own evolutionary thing independent of your biochemistry textbook based on yeast and plants and animals. You know, I've been doing my own thing for 150 million years. So you could tell me what I'm supposed to do, but I'm finding my own way. And when you look at diatom genomes, you see that they're quite a mixed bag of genes. This is a Phaodactylin genome. Chris Bowler was also one of your speakers in this course, I believe. And you see that the diatoms just have a mishmash of genes. And so if you really want to learn something unique, maybe this omic approach is the way to go. And my colleague, Andy Allen, did exactly that. I believe you had his wife as part of your seminar series as well. So I'm in amazing company here. So he went to Paris to work with Chris Bowler back in 2004, I think it was, or 5. And he basically looked at a whole transcriptome under low and high iron, and he found this interesting protein that he named iron stress-induced protein 2. Later, he made some genetic constructs where it's you could find the localization, found it was localized to the cell surface. It was one of the most responsive transcripts in his transcriptome. What does it do? So fast forward a couple of years, he had an amazing student, Jeff McQuaid, who started to dig in and noticed that this ISIP2 looked a lot like, at least from a 30,000 foot perspective, or excuse me, 9,700 meters. Thank you, Ronald Reagan, for not letting the United States adopt the metric system. And so he noticed from that 9,700 meters or so that the human transferrin and the diatom protein had similar location of particular residues that are involved in binding iron or carbonate. And that's a little bit of wishy-washy kind of line of evidence in and of itself. He also noticed, though, that in transferrin in the bloodstream, it's floating around. If you had this floating around in the ocean, you would lose it. So in fact, this protein has a transmembrane domain. It's anchored at the cell surface. Then he did some experiments with an endocytosis inhibitor. So remember, transferrin requires endocytosis. This is, here is a picture of cell membrane in Phaodactylin being labeled, and you're adding iron to a starved cell, and then you get these endosomes loaded with extracellular cell surface membrane. And then he did some work with our red fluorescent protein chimera with ISIP2 and saw internalization of that. And the endocytosis inhibitor knocked down iron uptake in this particular diatom. Then it gets really crazy. He knocked out the gene he's from one of the few labs that can do this, knocked out the gene, saw no iron uptake activity using a radioisotope. Then he complemented that strain with copies of the genes from humans and restored activity. That is crazy because the evolutionary history of humans and transferrin, in fact, is very different from diatoms. It looks like this actually evolved. This is a case of convergent evolution. But this tells me that the tools that are, ISIP2 doesn't do this all by itself. It has to engage other machinery for the endocytosis to happen. It's just the iron binder. So the fact that you could put back a human protein and then have this work suggests that a lot of the machinery is very, very similar. Very fascinating stuff. And the uptake in Phaodactylin depends on iron and carbonate. And so this is some of the data looking at the uptake rate as a function of carbonate at one iron level, a high iron level, and at a low iron level. And you can't see it from this graph, but the low iron level, the slope is still going up. More carbonate means more uptake. And so we were able to solve this data and figure out what are the rate constants. Can we describe the uptake rate as a first order process with respect to iron concentration, which is what you'd expect, double the iron, double the uptake, and as a first order rate constant with carbonate. Double the carbonate, double the uptake. So it's not the 0.78 and 0.001 here aren't the important values. What's important is that this is the iron and carbonate concentrations raised to the first power. So we were able to deconvolute and calculate the uptake rates of iron based on this pathway as a function of iron at one carbon at collapsing the carbonate concentration, if you will, and as a function of carbonate collapsing the iron concentration. And so I will be happy to bore anybody with this slide. I was able to do this modeling in Excel. It didn't require a supercomputer. It was a pretty straightforward iterative process, one that Rachel and I learned in chemical oceanography in, again, a year I won't divulge. So we applied our chemical oceanography tool here to figure out something about uptake kinetics and diatoms. And so we were able to model this to best understand that with this model, we see that the R squared is 0.999 here, which is a predicted uptake rate based on the calculated rate constants. So it's a first order rate process with respect to carbonate concentration, as well as with respect to iron concentration. So the empirically determined rate is here, the predicted rate is here, and our model is a very good fit. So where am I going with this, and why am I telling you about the model? Well, most countries in the world, I don't live in one of them, believe in that global warming is happening due to carbon dioxide emissions. And I should say that most of, get off the wrong track here. So business as usual CO2 emissions are shown here. This is the predicted concentrations in the atmosphere. If we keep on doing what we're doing with an annual increased rate of CO2 emissions, or if we listen to various strategies to curb our CO2 emissions to keep them at some rate, atmospheric CO2 will still increase, but at a much lower amount. So business as usual CO2 emissions have the untoward effect of continuing to acidify the oceans. And that's because as more CO2 gets in the water, it converts to carbonic acid. The carbonic acid reacts with carbonate and does two things. It decreases the carbonate pool and lowers the pH. So carbonate in seawater acts as a buffer to help slow down the decrease in pH, but it can't avoid the decrease in pH. So what we know is that if we keep on doing what we're doing, the pH of the oceans will lower enough to decrease. So this is an equilibrium plot called the Biuram Plot. Did I pronounce that right? Okay.
I thought it was Biuram.
Well, I thought the J, you know, I was trying to be Scandinavian in my,
Okay, well,
maybe I messed it up.
You got to wrap it up.
Okay, very good. So we see that at lower pH, carbonate disappears, bicarbonate grows into the system. And so this means that this pathway, if it's super important for iron uptake, and iron is super important for diatoms to fix carbon, as we pump more CO2 out, this pathway is going to suffer. Whether it, you know, so ceteris paribus, all other things equal, this means that the productivity in iron limited regions of the ocean could decline because the cells can't get at the iron that might be there. Summary, our work on ferric permeases suggests that reduction might not be required in our centric diatom T. pseudonana. The iron carbonate convergent evolution is really fascinating to me. Transferrins or phytotransferrins are widespread, suggesting that there's a positive feedback loop of negative consequence from ocean acidification on the biogeochemistry of iron. And these omic approaches really improve our understandings in systems where we don't, you know, where the sky is the limit as long as free energy yields are in the right direction. You know, like I said, these organisms didn't get a copy of the latest version of biochemistry. So we have to keep our minds open to what new discoveries are out there. And with that, I'll take any questions.